Welcome to G Fun Facts Online, where we explore the science behind the world around us. Have you ever been to a lake or a pond, dropped a line in the water, and pulled out a perfect trout or a feisty bass? Have you ever wondered how all those fish got there in the first place? It's often not just a happy accident of nature. In many cases, it's the result of a complex, high-stakes scientific process called fisheries restocking. This isn't just about dumping fish into water. It's a sophisticated endeavor that balances ecology, genetics, and human needs. Today, we're going to take a deep dive into the intricate science of replenishing our inland waters. Let's get started. The reasons we restock are incredibly diverse, and each has its own scientific playbook. Historically, it all began in the United States, back in the 1800s, primarily to make sport fishing more exciting. That goal, known as recreational enhancement, is still a huge driver today. Increasing the number of catchable fish like trout, bass, and salmon boosts local economies through tourism, fishing licenses, and gear sales. But the mission has expanded far beyond just sport. Conservation and restoration are now critical objectives. When wild fish populations are decimated by things like pollution, habitat loss, or overfishing, stocking can be a lifeline. Scientists introduce hatchery-reared individuals of threatened or endangered species to help rebuild populations that can eventually sustain themselves. This is usually a temporary fix, designed to work alongside efforts to clean up the environment and restore natural habitats. In other parts of the world, especially Asia, restocking is a key strategy for food security. This approach, called culture-based fisheries, uses regular stocking in lakes and reservoirs to maintain a steady supply of food fish, particularly where natural reproduction can't keep up with the demand. Finally, there's mitigation. Sometimes, human activities cause unavoidable environmental damage. Building a dam might block a salmon's migration route, or a pollution spill could wipe out a local fish population. In these cases, stocking is used to compensate for the damage and help the ecosystem recover. So once you know why you're stocking, the next massive question is, what fish do you choose? This decision is critical and depends entirely on the goals and the specific environment. Trout, including rainbow, brown, and brook trout, are superstars for stocking in cooler waters. They're tough, anglers love them, and rainbow trout especially can handle some temperature changes, although they are happiest in water below 70 degrees Fahrenheit or about 21 Celsius. If you're looking to manage an entire pond ecosystem, you might bring in a top predator like the largemouth bass. They are often stocked to control populations of smaller fish, like bluegill, and are a prized catch in trophy fisheries. But every predator needs prey. That's where sunfish, like bluegill and red deer sunfish, come in. Bluegills are stocked as a primary food source for largemouth bass, and they reproduce very quickly. There is even a subspecies called the copper-nosed bluegill, known for its aggressive appetite and fast growth, often reaching over a pound in well-managed ponds. The red deer sunfish, also called a shellcracker, is a specialist. It mostly eats snails and other mollusks, so it doesn't compete much with bluegill for food, creating a more balanced food web. Then you have species like channel catfish, which add variety and can thrive in many different environments. And for a real scientific twist, some programs use hybrid fish. A common one is the hybrid sunfish, a cross between a green sunfish and a bluegill. These hybrids have a very high percentage of males, which means they don't reproduce much. All their energy goes into growing big and fast, which is great for anglers. The journey for these fish begins long before they see a lake or a river. It starts in a hatchery, which is essentially a high-tech fish factory. Modern hatcheries are incredibly sophisticated, with different ponds or tanks for each life stage, from tiny fry to nursery ponds for juveniles, and finally, production ponds for market-sized fish. Biosecurity is a top priority. To prevent the spread of disease, these facilities have strict protocols, like footbaths and quarantine ponds for new arrivals. At the heart of a good hatchery is broodstock management. This is all about maintaining the genetic health of the fish that will produce the next generation. Managers keep detailed family trees or pedigrees to prevent inbreeding and ensure a balanced sex ratio during spawning. The health of these parent fish is paramount, as their nutrition directly impacts the quality of their eggs and sperm, and ultimately, the survival of their offspring. Once the fish are ready, they face one of the most stressful periods of their lives: transportation. They're moved from the hatchery to the stocking site in large tanks with aeration systems, or in sealed plastic bags filled with pure oxygen. Upon arrival, the most critical step is acclimation. You can't just toss them into the new water. The temperature shock alone could kill them. The rule of thumb is to gradually equalize the temperature, changing it no more than about 7 or 8 degrees Fahrenheit up, or 5 degrees down per half hour. For fish in bags, this often means floating the sealed bags on the surface of the pond for about 30 minutes. An interesting pro tip: managers are often advised not to mix the pond water with the transport water in the bag. This is because a lot of carbon dioxide builds up in the bag during transport. Opening the bag and mixing in new water can cause a sudden, dangerous shift in pH that can harm the fish. Now we get to one of the biggest scientific hurdles in all of restocking: genetics. Introducing hatchery-reared fish into a wild environment is a genetic minefield. When hatchery fish interbreed with wild ones, it leads to something called genetic introgression, the mixing of their gene pools. This can be devastating. Wild populations have spent generations evolving specific adaptations to their local environment. Mixing in genes from hatchery fish can dilute these adaptations, leading to a phenomenon called outbreeding depression. This means the hybrid offspring are less fit, with lower survival and reproductive rates. How does this happen? The genetic makeup of hatchery fish can be altered in a few ways. First, there's the founder effect. Hatcheries are often started with a small number of founder fish, which might not represent the full genetic diversity of the original wild population. Second, there's inbreeding. In the confined space of a hatchery, it's easy for closely related fish to breed, which can lead to reduced fitness. And finally, there's a powerful force called domestication selection. A hatchery is a safe, predictable environment. Fish that do well there, maybe they're less fearful or more aggressive eaters, are the ones that survive and reproduce. But those same traits can be a death sentence in the wild. Shockingly, studies have shown that even when hatchery fish come from local wild parents, their reproductive fitness can plummet with each generation they spend in captivity. To avoid these genetic pitfalls, responsible programs use broodstock from the local wild population whenever possible and limit the number of generations fish are reared in the hatchery. Constant genetic monitoring of both the hatchery and wild populations is essential. Beyond genetics, restocking walks an ecological tightrope. Introducing a huge number of new fish can send shockwaves through the entire aquatic ecosystem. Stocked fish can compete with native species for food and habitat. This is especially true if the stocked fish are aggressive predators or are introduced in very high numbers. In a strange twist, hatchery-reared fish are sometimes more easily caught by anglers. This can create a false sense of a healthy fishery, masking a decline in the wild population that's being outcompeted. Disease is another major risk. The high-density conditions in hatcheries can be a breeding ground for diseases and parasites. Releasing these fish risks transmitting pathogens like infectious hematopoietic necrosis, or IHN, to wild populations with potentially devastating results. The impact can even change the entire food web in what's known as a trophic cascade. For instance, some studies have shown that stocked trout can monopolize terrestrial insects that fall into the water. This forces native fish to switch to different, perhaps less nutritious, food sources, affecting their growth and survival. With all these risks, how do scientists measure success? This requires a robust, scientific monitoring plan, which is a critical, but sometimes overlooked, part of the process. Before a single fish goes into the water, a pre-stocking assessment is done. A key part of this is determining the water body's carrying capacity, that is, the maximum number of fish the ecosystem can support without negative effects. One way to estimate this is through a bait-based estimation, where scientists calculate potential fish yield based on the abundance of natural food like phytoplankton and zooplankton. A more practical approach is a density index, a rule of thumb based on fish length. For trout, for example, a common index suggests the environment can support between half a pound to 1 pound of fish per cubic foot for every inch of the fish's length. The most sophisticated tools are bioenergetics models, which use complex calculations to model fish consumption rates and predict how stocking will affect fish growth based on available prey. Once the fish are released, the post-stocking monitoring begins. A classic method is the mark recapture study. Scientists mark a sample of fish before release, then later capture another sample. The proportion of marked fish in the second sample allows them to estimate the total population size. For a wider view, they might use hydroacoustic surveys. This is essentially fish-finding sonar that can map the abundance, biomass, and distribution of fish over a large area without ever touching a fish. It's often done at night when fish are more spread out. To understand the human side, managers use creel surveys, where they interview anglers to collect data on what they're catching, the size of the fish, and how much time they spent fishing. This gives direct feedback on the fishery's performance from the user's perspective. And perhaps the most exciting new tool in the box is environmental DNA, or eDNA. Fish constantly shed DNA into the water from their skin, scales, and waste. Scientists can now take a simple water sample, analyze it for these genetic traces, and determine which species are present. It's a powerful, non-invasive way to monitor both native and stocked fish communities. All of this incredible data is then fed into quantitative models. To truly evaluate success, bioeconomic models are particularly interesting. They integrate biological data, like fish growth rates, with economic data, like the cost of fishing, to assess the overall performance and profitability of the fishery. Survival rate models use data from tagged fish to estimate how many of the released fish are surviving and what factors might be affecting them. So as you can see, restocking a lake or a river is so much more than just a bucket of fish. It is a sophisticated scientific ballet requiring a holistic approach that considers clear objectives, genetic consequences, and the delicate balance of the entire ecosystem. As human pressures on our inland waters continue to mount, the need for these scientifically sound, responsible restocking practices is more critical than ever. By using a strategic and adaptive approach, we can use restocking as a powerful tool to enhance our aquatic resources while protecting the intricate web of life they support. Thanks for tuning in to G Fun Facts Online. Join us next time for another deep dive into the science that shapes our world.